Well, hello, Bio30s. We are going to talk today a little bit about protein synthesis. So I have for us here our little notes guide, and hopefully you've watched some videos and read through the slides. Now, in order to do this booklet, you need one more piece of information, and I have posted this for you guys. I call it the mRNA magic chart, and it shows you a list of the messenger mRNA codons and their amino acids. So you need this handout in order to complete this handout. So let's talk a little bit about protein synthesis. So we are building proteins, making proteins. Protein synthesis means building or making proteins. Proteins, of course, are made of amino acids, and they're the most versatile things in your body, right? They have a variety of functions. So we know that amino acids string together and they make this long polymer called a protein. And proteins are used in a whole bunch of things in your body, like hemoglobin is a protein for transporting oxygen in your blood. And another example of a protein, enzymes are proteins, right? And so they catalyze all these reactions in your body. So what is the genetic code and how does this relate to protein synthesis? Well, our genetic code is our DNA, right? The sequence, the order, the bases, the letters on the DNA. So a DNA sequence that might code for protein might be like ATA, CCG. So if you remember, DNA is this massive double-stranded molecule, and we draw it kind of conceptually. It looks something like this, and it looks like a twisted staircase, a spiral staircase, or a twisted ladder. So all of these DNA bases, this is our genetic code, right? And we have millions of bases on the chromosomes in our DNA. And so we only take a small section of this. So maybe just this section makes up a particular gene, and that particular gene is going to code for a protein. So one gene, one protein, and then that protein is going to do something in your body. So maybe it's an enzyme, or maybe it's hemoglobin, or maybe it is going to be an allele that is expressed as the color blue for your eyes. Okay, and so these are how DNA genes are related to, well, they are the genetic code and they are the instructions for making a protein. So the structure of a protein, the DNA, the bases, the genetic code is this set of instructions for how to make proteins. Enzymes are proteins. And so these DNA letters is the template, the original instructions, the original information to make a protein. One gene, one snippet of DNA is going to make one protein, one enzyme. So here we have what we call the central dogma. So this is found in every type of cell. It's universal, whether we're talking bacteria or viruses or humans or pigs. This is the two steps of protein synthesis, making a protein, expressing a gene, taking that genetic information and ultimately making a protein. So step one is called transcription. That's where the double-stranded DNA is made into the single-stranded RNA molecule. And then in the second step, the RNA is made into an amino acid. And then the amino acids are put together in a string. We call that string a protein. So there's these two steps. Transcription is first, translation is next. So both of these steps are the steps of making a protein, protein synthesis. So transcription is happening where DNA is such a big molecule, it can't leave the nucleus. So you have to make a messenger. That messenger, mRNA, M stands for messenger. So now this little mRNA can leave the nucleus and take the message out to the cytoplasm, and that's where the protein is actually made. So the transcription is happening in the nucleus. DNA is being made into a different language, the language of mRNA. And then the second step of protein synthesis, this is translation. So the mRNA is read by a ribosome. So this is happening out in the cytoplasm. That's where ribosomes are. And then we have another type of RNA, a transfer RNA. He's the delivery guy, and he's going to bring in an amino acid. We make this strand, this sequence, this chain of amino acids, and that is a protein. And so one gene, one segment of DNA is going to make one protein. So this might be the part of the DNA that gets transcribed into the RNA. RNA is read and it gets made into a protein. So one gene is expressed as a protein. And again, that may be an enzyme, it might be hemoglobin, it could be a variety of different things. Immunity, that's proteins. There's all sorts of things that proteins do in your body. Okay, so this question, when you are asked to identify amino acids that correspond with mRNA codons, this is where you got to grab your magic chart out. You have these list of mRNAs, and you're going to determine the 3-letter mRNA codon that makes a particular amino acid. Okay, so if you just turn to your magic chart, and so I'm going to pull up my magic chart, and here's the first 3-letter AGA. These are the mRNA letters, so we call this an mRNA codon. So there was a DNA sequence which got transcribed into mRNA. So how do we use this table? So what is the first base here? A is the first base. So we go and we look at A. Okay, what is the second base? G is our second base. So now we look, here's A and here's G. So we're finding this box here. And then what's the third base? AGA. So we have ourselves, this is the mRNA codon. What amino acid does it call for? It calls for the amino acid arginine. Okay, and so that's how you use your magic chart. Now, you'll also notice on your magic chart that there are several different mRNA codons that don't code for an amino acid. In fact, what they code for, they're called stop codons, because they're found at the end of a long string of amino acids, and they stop the production of the protein. So maybe it goes like leucine, arginine, serine, tyrosine, stop. Well, that's it. That's all the amino acids you're going to get in that particular protein. Okay, the stop means you're done. You're done transcribing, you're done translating, you have created a particular amino acid chain, which we know as a protein. So you'll also notice on that chart that some amino acids have more than one codon. So if you just saw, and I'm going to go back to arginine there for a second, there was 6 different codons for that same arginine, 6 different ways that you can make that same arginine amino acid. And you'll notice that for them, CG is the same. CG is the same. It's just that third base that is different, right? There's a little bit of wiggle room on that third base. There's still, all 3 of them are coding for this arginine. And then here's 2 more, AGA, AGG. So there's more than one codon in order to get the exact same amino acid. We say that the genetic code is redundant, right? There's more than one way to get the same amino acid. So it's that last base, that last letter, that's going to be different, and that's why we call it redundant. Okay, and so just a reminder, you'll see that's not the only one. So there's 2 for arginine here, and there's 4 more here. So another example, leucine, right? There's 6 different ways to make leucine, and it's that third base, that third letter in the mRNA codon is what is going to be different from them. Okay, so next up, let's take a look at a couple of questions here, and you can read through here, watch the videos, use your textbook. Here is a DNA sequence. Another word for this is DNA triplets. And so they've given us the DNA letters, and they're asking us what is the mRNA. So we have to transcribe from our DNA to our mRNA. So all we do is we use our matching, our complementary rule. If it's a T on DNA, it's an A on mRNA. Now remember, there's no T in mRNA, so instead there's going to be a U. So then once you have transcribed and you've got your mRNA sequence, your codons, then you're going to use your magic chart, and you're going to look at these 3 letters at a time, and using your magic chart, now you go and you decide, okay, so I've got AUG, that was my first 3 letters. AUG, well, here he is, methionine. That will be the first amino acid in this particular strand, or in this particular chain that is going to make a protein, okay? And so then we would do that for the next 3, and the next 3, and the next 3, okay? And so that's how we are going to transcribe from DNA to mRNA and then translate. So again, you use your magic mRNA chart to go from mRNA to amino acids. So you look it up, methionine, proline, etc., etc. Okay, so using your chart, you can build an amino acid sequence, a protein. And then the next question is showing you've got to do the exact opposite. You've got to work your way backwards. So we've got methionine and lysine, histidine, tryptophan. So you look up each of these on your magic chart. So methionine, we just found that one. And when we looked it up on the mRNA, it was AUG. And then we go backwards one more step. What's the gene sequence? Well, here we're going one more step back to the DNA. What's the 3-letter DNA triplet? Well, it's going to be TAC. Okay, and then we're going to look up lysine. And if we look up lysine, we see that it is A, and then either A or G. So now we work backwards. It's going to be TT, T, or C. All right, so again, this is showing us going forward, our protein synthesis, translating and transcribing. And this is just another skill. You have to be able to do this both forwards and backwards. If I give you the amino acid sequence, work backwards using your magic chart to the mRNA, and then backwards from that to the gene sequence, the DNA. So again, watch the videos for this week, read through the PowerPoint, read through your textbook questions. And if you have any questions for me, don't hesitate to email michelle.ledeen@ghsd75.ca. Take care. Talk later.